Introduce you first. So welcome to Mathematical Biology Seminar. Today we have Jonathan Rubin as a speaker. He's a professor and a chair at the Department of Mathematics at University of Pittsburgh. He will share with us his research today. Jonathan.
Great, so let's share. Okay, so I guess you see my screen? You see Pittsburgh?
Yes.
Okay, if you see me glancing off to the right, that's because that's where I have your faces, and it's nice to see faces once in a while in the talk. So yeah, so today I'm going to talk about decision making, and so I picked up this little video from YouTube as a sort of demo of what I'm talking about. And so this is an interactive exercise. Okay, so first let me get there. Okay, so this is going to be an example of what people call perceptual decision making. And this is what's known as a coherent motion task, as you can see. So when I click the play button, these dots are going to start moving around. They're going to have drift, not strictly left-right, but up-down as well. But there is a dominant movement direction. So they're more coherently moving in one direction than the other. And your job is to decide whether that is to the left or to the right. And so I was told you have access to the chat. So when you know that when you have your answer, then as soon as you have a reasonable level of confidence, please go ahead and just type an L or an R in the chat. Okay, so let's try. All right, and now let's see, we have maybe fewer than half have answered. So now I'm gonna go into a forced choice element where you have 10 seconds left. If you haven't answered, please answer in the next 10 seconds. Three, two, one. Okay, maybe it's possible that only panelists can do the chat. It seems all the panelists have answered. So let me stop this. The point of this isn't whether I'm not going to tell you whether it was left or right. That's not the point. The point is, in the chat we have 5 lefts and 2 rights. Okay, and several came in at 12:16, but they were scattered very broadly in time throughout that minute of response. And you can see I played the video for 1 minute and 10 seconds, and we had a couple of responses quite early in that, and a couple towards the end, and one person, like as I was doing my countdown, it finally came in. Okay, so that is what we call reaction time distribution in this task. And we also, as I said, have a distribution of responses. So, if let's assume that the dominant motion was to the left, that's also how I perceived it, then we would have, basically like a 3/4 accuracy rate, since that's, the number of trials here that led to a left, where I'm considering each person who responded in the chat as a separate trial. Okay, so if you think about how you did this task, you gathered information, right? And eventually you felt like you had enough confidence to make a decision. And maybe you wanted to stop paying attention, so you answered quickly. Maybe, you thought I might give a prize to the first correct answer, so you answered quickly. Maybe you really, really, really didn't want to get it wrong and be embarrassed in front of everyone, and now your reputation's ruined, and so you took your time. So depending on your incentive and your priorities, you might, try to do this task differently. Okay, so we can think about this process. I just described it in these vague terms, but we can try to think about this more mathematically, right? And so to do this to think about this mathematically, we should try to phrase a more precise question that we want to ask about how this works. And so one way to think about this process is what I've called the decision problem here, right? So let's imagine that we have readouts sort of corresponding to these some visual system component being driven to register leftward movement, and another such component responding to rightward movement. Okay, so given that you're staring at this stimulus whose statistics don't change over time, these are going to have these mean activity levels, but there are also going to be fluctuations, right? Any system like this is going to have fluctuations, so it's not like you can just be sure that whatever activity level is higher is going to show up in your readouts. And then given that information, you want to determine, which of these two hypotheses holds. Okay, so that's one way to sort of start formalizing what's going on in this situation. Okay, and then you can also, try to phrase this as an optimization problem, right? So given that you have this setup, we can ask, what choice strategy yields the best decisions, right? That would be a reasonable mathematical problem to study about this. So in order to do this, look at this as an optimality problem, we need a definition of best. So I wrote down a couple of possibilities here, right? So one would be that we fix the response time, and we're going to imagine we're doing this over multiple trials perhaps, but we have a certain time in which we need to respond, and we want to get the lowest expected error rate within that time. Another way to look at this is to fix the acceptable error rate and figure out what sort of response strategy would give you the shortest expected response time to achieve that error rate, okay, over multiple trials. Okay, and this problem was already solved quite a while ago. Okay, and so the answer is if you want to come up with the best strategy in these terms, these A and B terms, what you should do is use what's called the sequential probability ratio test to make your decisions. Okay, so imagine that you're collecting samples, so you're looking at the dot motion for a little while, and based on your first time increment, you determine there's a certain probability in that time increment y1 that, let's say, 1 is left. So there was leftward movement, and another probability that there was rightward movement, so that gives you 1 ratio. And then you build this product over time. So over your next sample, you get probabilities, your next sample, you get probabilities, and so on. Okay, and then you have some bounds. Okay, so one of the bounds is bigger than 1, one is less than 1, and when your ratio eventually exceeds the corresponding bound, you either choose, option 1 or option 2. Whoops. Okay. Equivalently, you can take logs and think of this as, it's a more natural way to think of it is to take sums of these logs of the probability ratios over each increment of evidence that you've collected. Okay, and likewise take the logs of the bounds, and then you commit to your decision when the ratio escapes the bounds. Okay, so this is the sort of best approach to this problem algorithmically. Okay, and it turns out when you take the time between these samples to 0, right? Take a limit as the discrete time steps here go to 0. This exactly corresponds to what's known as the drift diffusion model. Okay, so this is a really nice, simple stochastic decision-making model. Okay, you can see it represented as a stochastic ODE here. In the DDM framework, we have several parameters. Okay, so we have a drift rate, right, this deterministic part. Okay, and in the optimal setting, what you should do is choose this. If you want to maximize your reward rate, you should choose this in this way. Okay, this is what shows up from this sequential test that I mentioned on the previous slide. Okay, so in terms of this log of probability ratio. Okay, so that gives you this drift. You have boundaries Okay, and you're going to decide when your drifting particle hits a boundary. Okay, so if it hits an upper boundary, then you're picking one choice, and if it hits a lower boundary, then you're picking the other choice. Okay, there's a noise intensity, okay, and a bias. Okay, so there's no reason that we have to start at 0. We might already have a bias based on past learning or something towards one of our options. Okay, and it's a really nice way to think about this process of evidence accumulation, and it's really nice mathematically, right? It's easy enough to deal with, so we know at least we can write down the probability density of positions of a particle diffusing under this rule, well, evolving under this rule, and it looks like this if we start from 0 bias. Okay, it's just normal around the drift. Okay, and then from there we can derive an escape time distribution. So that was done a while ago. And mathematically this is actually pretty nice, even though it's an infinite sum, because if you define this ratio of bias to threshold, then you can express this in this recurrent way. Okay, so the escape time that you derive is this infinite sum, but you can describe it in this recurrent way. And making use of that, there's a nice 2009 paper that comes up with a couple of different error bounds. So one is based on sort of a small time truncation, and the other is based on a large time truncation, where you can justify that a certain number of terms are relevant. And you can see sort of, in order to get an error below a fixed bound, this is how many terms you need in your sum, depending on which sort of approximation you take. The details of this aren't precise, aren't important for the talk. I'm just giving some background on sort of what's been done in this area. Okay, and there's a more recent nice work, for example, this SIAM review paper I would recommend to anyone really interested in decision making, where the generalization here is where there's a probability that the evidence is changing at each time step. So maybe the overall drift direction, dominant drift direction changes. Okay, so that's some background on sort of the classic directions in thinking about a problem like this. Okay. Sorry, this is a visualization of the drift diffusion model, and this is going to show up throughout the talk. So I just want to orient you here, right? So this is what I was talking about, where we have these, for each trial, you can think of a particle moving according to the DDM, and eventually it may hit a boundary, and that's when you commit to that choice. And you see that you can end up with reaction time distributions which depend on all of these parameters. So I'm focusing on 4 parameters here. There's a little inconsistency. So we have a certain delay before we start collecting information. Okay, so that corresponds to a mechanism to sort of translate our reaction time distribution without changing its shape. And we can have a bias, drift rate, and this boundary separation. And we ended up, for the work I'm describing, using z times a as our bias. Okay, so 0 would still be no bias, but it's this sort of scaling. And that was a case where, not because this gives you some advantage, but more like we had done the work and realized that's what we had done, and it seemed silly to redo everything, just changing this back to z, knowing the results would be the same. So yeah, in any case, what's important for what I'm going to be saying is, that as I mentioned, we can change parameters in this model, and that'll change the features of the, how accurate we are, how often we pick the correct choice, and what our reaction time distributions look like. Okay, so as I mentioned, the onset parameter, which I'm going to actually call t, gives us a pure translation. Okay, changing the drift rate, which would correspond to changing how strong the evidence is for one option or another, would change the reaction time distribution in a certain way. And changing the boundary height changes the distribution in a different way. Okay, so we can sort of favor speed by lowering the boundary height, or by speeding up the drift rate, but those are going to give us different reaction time distributions. Or to phrase it differently, if we start from a fixed V and A, if we slow down V or increase A, those are two different ways to sort of aim for accuracy and slow down our decisions, but they'll give us different reaction time distributions. So based on that idea, we can kind of think of the DDM as giving us a parametrization of a decision policy space. Right, so we have these, it's really a four-dimensional space, and our decision policy is based on our accuracy and our reaction times, right? And so we can ask, are we favoring speed? Are we favoring accuracy? And here, looking at this 2D view, forgetting about the other two parameters, but by manipulating this speed of evidence accumulation and boundary height, we can sort of move to a high accuracy regime, low accuracy regime, right? We can change our reaction time. And so we get these sort of two different levers of control that allow us to pick whether we want to be in a relatively fast regime where we're exploiting, so we're sort of always sticking with the high reward probability outcome, or we could be in a more exploratory mode where we switch back and forth quite a bit. We make fast decisions, but we're not so worried about accuracy. And it's important to have in mind that all of this makes the most sense when we're in a situation where rewards or what answer is correct actually changes, is not deterministic, right? So in other words, in the if we think of a two-choice task where we have two halls to go down instead of the moving dots, okay, and we choose sort of the correct hallway based on whatever evidence is part of the experiment, maybe we have a 75% chance of getting a reward. Okay, if we choose
Sorry, I think Paul is raising a hand.
Yeah, if we choose the incorrect hallway, then we still have maybe a 25% chance of getting a reward. Okay, so that's why we might, that it's important to think in those sort of terms so that it isn't like a situation where, yeah, it's completely obvious that there's one right answer and we should just always, pick that one. So, Paul, question.
Yes, you're saying the drift rate is something you can control, but isn't the drift rate the signal? So if you just increase the drift rate without changing noise, you're improving the signal to noise, which in general you can't do because you're given as general signal and noise.
So I am thinking of this as, this is my algorithm for making a decision. So I am controlling my DDM parameters, right? So,
but if you scale the amplitudes, the noise will scale, right?
So I'm observing this process, right? And so I'm making these estimates of the probability of left, the probability of right. And then based on those estimates, I'm going to choose my drift rate.
But I had a similar question, but isn't that true that the variance, I think I saw that in your equation, the variance is related to the drift? The diffusion coefficient?
That's the optimal case, right? So in other words,
that's not what you're using. Okay.
Want to connect this, right? So, right, this work has shown that in order to get sort of the best performance in these couple of in these couple of notions of optimal, you should use this criteria for making your decisions. And then if you put that in the context of the DDM, that tells you, okay, the drift rate is determined by the signal, as Paul put it, and the noise intensity is related to the drift rate. Right? That would be optimal. Okay, but when
But that's not what you're using.
when I go here, what I'm going to do now is not necessarily start from optimal, right? I'm going to say I have the freedom to choose my drift rate, choose my bias, choose these 4 parameters. Okay. And if it's really, really important to me not to make mistakes, that might lower my reward rate, but at least my accuracy will be really high, right? So I have the freedom to deviate from optimal if my definition of optimal is different, isn't maximal reward rate, or if I don't know the sequential probability ratio test result, right? So, yeah. Is that okay?
Are you saying that in principle, even if the evidence is pointing down, you could choose a drift rate that's up, and now you're going to show that is optimal to have it proportional to the...
Yeah, but even I might choose a drift rate that's up, but not the optimal drift rate. Right? It might be a shallower slope than optimal. So you can imagine that's the case in many situations, right? I mean, if I have a brand new task, I might not, or I might have a different incentive than maximizing reward rate, as I said, or I might, I mean, I might not know, what is the way to maximize reward rate, even though that's what I'm after, right?
Okay.
So,
and just to finally clarify, so you're not allowing the noise to be varied, so you're assuming everything is in units of the noise, and that's kind of the dimensionality.
Yeah.
And related to that, also I had a second question, but do you ever look at the variance of the error rate and response time?
Yeah, so when I say that, varying these different parameters changes the distribution, it's changing not only the mean, but all the moments of the distribution. And one can, compute some of these things analytically for the regular, drift diffusion model that I showed. Right? You can if you start with no bias and just fix these parameters, you know that you get a normal distribution of positions, and from there you get this escape time distribution, right? And you can, so that is the reaction time distribution, and you can look at its features at least up to some approximation. Okay, so the drift diffusion model, the main takeaways here are, it's connected to optimal decision making. Right? That we know there's a way to change, to tune the parameters that would be optimal. Okay. More generally, by varying the parameters freely, we can move around in this sort of decision policy space and aim for the priorities that are important to us, or deal with some sort of change in conditions, right? Or some scenario where we're actually learning about the reward contingencies. Okay, and it's really intuitive to think about this, I would say, algorithmically, right? Because it matches what we think about when we think about making this choice, where we're collecting evidence and so on, until we feel like we've accumulated enough to hit some confidence threshold. Okay. So if you believe, all of these attributes of the DDM are useful, and we believe that sort of our, animals' performance, tends to be optimal in some sense, then that leads to a natural biological problem. Okay, so up till now we were sort of looking at the mathematical way to think about this problem, but biologically, right, what's implementing this should be something neural, right? It's the nervous system where we take in whatever the evidence is, whether it's visual, auditory, olfactory, or whatever, and we know there are parts of the brain where this happens. And so if we think DDM is the right sort of decision-making framework, then we might imagine, there's some chamber in the brain, right, where we have diffusing particles that get tuned by the input, and that is how we, take advantage of this framework that can give us optimal decision making, or even better, tunable decision making to pick out our priorities. Okay, so then that would lead to the question of where in the brain is this, DDM-like activity happening. Okay, and so obviously there isn't literally going to be some chamber of diffusing particles, but there might be some circuits in the brain that act in this way. Okay, and so for the rest of this talk, I'm going to focus on a circuit that people have thought of as being related to this process, which is the network of the basal ganglia. Okay, so these are a connected network of several different subpopulations deep in the basement of the brain, right? That's where basal comes from. You can see some of their names over here with these colored regions, and you can see a little primitive wiring diagram linking some of them over here. Okay, and so one of the reasons that people like to think of the basal ganglia as relating to decision making, is that it is a site of release of this reward chemical dopamine. Okay, so probably even if you don't work in neuroscience, you've heard of dopamine, but this is what's called a neuromodulator. It's a chemical that gets released by certain cells. It's been shown to relate strongly to whether you get a reward for an action or not. And a lot of that happens in the basal ganglia. Okay, so that makes this a natural site to think about for decision making and learning from the rewards that you get. Okay, and so people have tried to do a mapping where they say, okay, we have this wiring diagram. We have these sort of high-level representations in this sort of drift framework. So for example, people have talked about like a believer-skeptic sort of competition where, to try to think of a neural implementation of DDM, you can imagine that there's a skeptic population that tries to prevent movement, a believer population that tries to promote movement or a decision, and they sort of compete, and that produces the trajectories here. You can impose on top of that an additional like braking process. So if something comes in that says, hold the boat, don't move, there's a predator watching you, whatever, that becomes its own sort of component that can push you away from threshold again, or push you towards a different stop threshold. And it turns out, you can sort of look at the wiring diagram of the basal ganglia and sort of make a mapping that says, yeah, if we sort of look at the basal ganglia as this feed-forward-like structure, where you get inputs from cortex, and you give outputs down here from this area called the GPI, okay, then some stream of connections here, if you look at which ones are excitatory and which ones are inhibitory, has the potential to promote a signal getting through, which would, in this framework, correspond to committing to a decision. And another chain of connections would tend to strengthen this inhibitory output, so prevent a signal from getting through. And then there's even a convenient sort of braking signal, which would make the inhibition coming out really strong and presumably shut everything down. Okay, so people have sort of done this, and that leads to some predictions about various ways that basal ganglia components could contribute to decision making. Okay, but for this project, we decided to flip things around. Okay, so the work that I'm focusing on here is several, recent papers that we've worked on, where we use what we call an upward mapping approach. Okay, so if downward is from conceptual or down to the brain, what we're doing upward with an upward mapping is going the other way, right? So what I have here is 2 copies of the basal ganglia wiring diagram, right? So there is some evidence Of course, things are never, extreme or idealized in the brain, but there is some evidence that there are these sort of parallel pathways through the basal ganglia. Okay, so all of the connections are there in each one of these pathways, but these pathways can be thought of as corresponding to action channels. Okay, so we might have a channel to move a specific limb or to do some specific movement, or there's lateralization, right? So we might have a sort of channel to favor left, a channel to favor right. Okay, so whatever. In this framework, let's just think of, we have a competition between two choices, and each one has its channel in the basal ganglia. Okay, so we can simulate this big network, which has these two channels, okay, receiving cortical input, leading to output, and we can impose a decision condition, okay, based on the output of the basal ganglia. And I'm not going to go into the details, but you can ask about that if you want them. And so we have these two channels sort of competing to reach the decision threshold. And whenever that happens, we record that as a decision and we start a new trial. Okay, now what's driving these decisions is going to be whatever activity we have up here, to begin with in the cortex, right? So we can think of the cortex as pulling together this perceptual information, or an internal version of this perceptual information where maybe we're just thinking about our experience in a similar situation and what we should do based on that. And then this cortical activity drives the corresponding channels and determines the decisions based on the decision condition. Okay, so whenever we have a decision, that gives us a corresponding reaction time. And over many decisions, we get many reaction times, and then we get a choice distribution, whether we've chosen A or B. Okay, and then for the upward mapping, we can then fit those outputs, we can think of that as data, and figure out what are the DDM parameters that best fit this data. Okay, because the DDM as well will produce a reaction time distribution and choice, distribution depending on what parameters we give it. Okay, and then the idea is to use that relationship to infer what are the contributions of the different basal ganglia components to this algorithm of decision making. Okay, so that's the idea, and I'll show you a little bit of how it works, and you can think about whether you think this is helpful or not. I'm very happy to discuss and take questions as we go or at the end. So to go into a little more detail on what we do, okay, so we're thinking again of some sort of two alternative force choice task. Okay, so you can think of the dot motion from the beginning of the talk, or you can think of a situation, maybe a rat or a mouse in a Y maze, or us in many situations in life, looking at a menu or whatever. But in this case, there's two items on the menu, and so we have to make a choice. And based on that choice, we may or may not get a reward. But for this part of the work, let's not worry about the rewards, okay? So we're going to make a choice. And to simulate this situation, as I said, we have our two basal ganglia action channels, and we populate these with spiking neurons. We have collections of spiking neurons in each one of these boxes, and we have the synaptic connections between all of these regions. Okay, and what we get dynamically, if we take the spikes from these regions and convert them into firing rates, we get simulations that look like this. Okay, so we set things up with cortical signals ramping up over time to the two channels, and we're not putting any bias towards one option or the other in place for now, because we're just trying to do this baseline exploration of this mapping. Okay, we should get the baseline in place before we go to more complicated situations. Okay, so what happens due to this cortical ramping? Okay, so we're getting these inputs coming to these what are called DSPNs and ISPNs. So these two different populations in the striatum, which is the input level of the cortex. Okay, so because of the cortical input, we should see upward ramping in the DSPNs and ISPNs. And then we have this extra inhibitory population that kind of helps keep these two in check. So that's also ramping up. All right, so then we have to look at what's happening downstream from these two populations. Okay, so one of them, the Ds, just projects directly to the basal ganglia output. Okay, and it's inhibitory. So it's trying to push down this output population, lower the firing of this population. Okay, meanwhile, coming through this other pathway, right, that's this arrow here, this side is trying to promote the activity of this population. Okay, so for a while, that balance, that ends up producing a balance. Right, so one side is trying to suppress it, one side is trying to promote it, and it's balanced. But eventually, the balance breaks, okay, which means the downstream target is freed from inhibition, and whichever side is freed first reaches decision threshold and a decision is made. Okay, so that's the way this network, this model works. Okay, so that's the first step, getting all of this set up and getting it tuned so it has reasonable firing rates in all the populations and sort of is driven by what's happening in the basal ganglia and not just by this direct projection from cortex to thalamus where the decision threshold is imposed. Okay, so hopefully that's clear. So then in the upward mapping, we sort of can think of iterating two loops. Okay, so in the outer loop, we're going to sample from our allowable distribution of weights of all these connections between the basal ganglia populations. Okay, and then once we pick the weights, we're going to run a whole bunch of decision trials. Okay, so a whole bunch of simulations that look like this. Here, yeah, many trials. I didn't specify how many. I guess here it was 300. Okay, and then from those 300 trials, right, we get results that look like this, right? We get, here we're just displaying the average firing rate in each population, 1 point per trial, right? So this axis is trials. We also get whether each trial led to a left or an A, let's say, or a B. Should be roughly 50-50. Whoops. And then we get a reaction time. Okay, and so we've tuned our network, so these are reasonable. And based on these pieces of information that we read out, that we fit, and that gives us a single best fitting set of DDM parameters. Okay, and then we iterate the outer loop again, and that's going to generate another single set of DDM parameters in this way. Okay, so here's an example of what we got when we did many iterations of the outer loop. Okay, so for each outer loop iteration, again, we got one set of DDM parameters, and we got one mean reaction time and percentage correct. Okay, and so here are the distributions of all 6 of those things. So the first 2, again, are coming from the basal ganglia. So percent of action A should be very much around, right around 50%. And then these are our DDM parameters. Okay, that best fit the output. All right, so we've explored manually the sort of range of reasonable weights that are present in the basal ganglia. Okay, so this is doing a lot of manipulation that could never be done experimentally. And by manipulating all of these things and effectively looking at how changes there map into changes in the DDM, we want to generate predictions about how changes in the basal ganglia would impact like decision policy. Okay, and that'll represent actual biological predictions about what might be the control mechanisms there in the basal ganglia that could impact how an animal makes decisions. Okay, so hopefully the logic is okay. Just good enough for at least a couple paper reviewers, so that's good. These are our results. Okay, so again, representing now the upward mapping in these as this mathematical mapping, right? So we're going from the weights that we're selecting to the parameters of the DDM. Okay, and it turns out if we try to look for a relationship there, it doesn't work very well. Okay, so one of the things we had to realize was that, you know, there's many different combinations of weights that can produce similar firing rates in the different populations because of all the different feedbacks and sort of balancing components. So we write this mapping as a composition of two pieces. Okay, so the first one is a mapping from the weights in these synapses, these connections, to the firing rates of all the different populations. And then the next component is a mapping from those rates to the DDM parameters. Okay, and then we could look for relationships embedded in either one of these mappings. And to do that, we used canonical correlation analysis. Whoops. Okay, so the idea of canonical correlation analysis is it's a method often used in statistics to look for directions of sort of common variation in two different data sets. Okay, so in our case, for example, we might have like 13 populations firing rates, so that gives us a 13-dimensional space times, you know, however many samples we have, however many trials we did. For each one of those trials, we have our one DDM parameter set. Okay, and then CCA looks for projections and vectors in the data space such that under those projections, you get the strongest alignment between the images of these vectors coming from the parameter space here on one side and the rate space here on the other side. Okay, so that's, this blog happened to have this nice visualization that I edited a little bit to you know, to put in our dimensions and so on, so you get an intuitive sense of what this is doing. Okay, so again, intuitively speaking, once we find these vectors, the idea is moving along one of these vectors, say in the rate space, will correspond to, in our data sample, a movement along the corresponding vector in the parameter space. Okay, so this is what we find. So we do this CCA, and we have to check how many pairs do we get that have a good enough correlation, a strong enough correlation in the image. Okay, in this case we found 3 when we looked at the relationship between the rates and the parameters. Okay, and one of the things that was important for getting meaningful results, I should have said, was instead of looking at the actual 13 rates themselves, we looked at sums of rates across corresponding populations in the two channels and differences in rates. Okay, and then we also have these two populations that are shared that we're not going to pay much attention to. Okay, so what we've done here is, you know, these are the vectors. Each one over here is 13-dimensional, over here is 4-dimensional, right? The strengths of the components in these vectors are indicated by the colors. Right, so green represents strongly positive here, this is strongly negative. And so the idea, for example, if we look along any one of these components, sort of moving along a direction with this collection of increases and decreases should correspond to moving along a direction in DDM parameter space with this collection of increases and decreases, and so on. Okay, so I'm guessing, you know, if you're staring at these two colored, you know, boxes, you may be taken by their beauty and the artistry here, but you may also be like saying, okay, you know, how in the world can I do anything with this, right? Because this is just this hodgepodge of colored strips. What in fact is much more useful is to impose these results on the actual wiring diagrams of the network and the DDM. right? So this is a useful representation of those same results. Right, so here we took those color codings. For example, the sum of DSPN in the first component is dark green, right? So here this is dark green. Right, so we took the color codings. We have a sum side and a difference side. We impose the corresponding color codings here so you can see what's going on in the network. And then in the DDM, we have the 4 different parameters. Okay, so the first component that comes out of this analysis is what we call the responsiveness component. And this is essentially just this sort of pure translation, right? So you can think of it as just when do we start collecting evidence, but effectively it's a lever for translating the reaction time distribution. Okay, so if for whatever reason we want to do that, okay, the neural changes that we should use to most effectively do that are indicated by the color coding here. Okay, and again, we shouldn't pay too much attention to these two FSI, CXI. They're shared and they're a little different than the rest. But the main colored components here are on the sum side. And one that stands out that I'll focus on is this DSPN, right, this dark green. So the idea here is this was this direct pathway, this sort of go pathway. And so what this is effectively doing is controlling how early the evidence accumulation begins, right? So increasing activity here corresponds to a later onset time of evidence accumulation and therefore a later reaction time distribution. Okay, secondly, we have this, what we call the pliancy ensemble. And it took us a long time to come up with the word pliancy. It's not one you would necessarily use every day, but pliancy is sort of willingness to be convinced. Right? It's related to skepticism, but skepticism has a more dimensional, it has a sort of direction associated with it, and pliancy is more neutral. Right? It's how willing are you to be convinced? And so what we can see here, right, is that we have this nice strong loading on the boundary height at or decision threshold. And sure enough, most of the action is showing up in the sums, and especially the dark regions here are associated with the indirect pathway. Okay, so as I've written down here, right, overall activity in the indirect SPNs correlates with this higher boundary height, whereas decreased activity here corresponds with a larger boundary height. Right, so basically this is an ensemble that allows you to sort of prioritize caution by changing activity in these ways. Okay, and then the third component shows up much more strongly on the differences side rather than the sum side. Okay, so we call this the choice component, and the idea is if you want to promote, a sort of faster drift rate and choice of this particular direction, a certain directionality in your decisions, then you should exaggerate the difference in firing in your direct channels, DSPN. You should reduce the difference in firing in the ISPN, and you should also make these other corresponding rate changes downstream. Okay. Yeah. So we really should think not just about the ISPNs have one effect, the DSPNs have another effect, but we should think about there are some effects related to overall activity levels. There are other effects related to differences between channels. And really the effects relate to a whole ensemble of rates, not just single rates, at these two sites in the basal ganglia. Okay, so that's kind of the main part of the talk. I said a lot more in the abstract, but I didn't want to feel like I needed to rush. I'm sort of tired of giving talks where I have to rush because I decided to cram in too much information. So hopefully the pace was reasonable, and these are the conclusions up till now, and then I'm going to just sort of mention a few other interesting things in a non-rushed way that shouldn't be overwhelming. Okay, so the method here is aimed at getting a hold of these control ensembles that modulate, these aspects of action selection, and we have these predictions. right? So for example, we predict that when you're confident and you really want to, push your decision policy in a confident direction, you actually want high overall ISPN activity, which is a little counter to what people usually say in this field. But you want a very channel-specific promotion of DSPN activity to make sure you make the right choice. Okay, and the other aspect of looking at this is, if you want to adjust your decision policy in a realistic way, not as a simulator, but as, using actual synaptic plasticity, then the most effective way to do that would be to do it in a way that somehow changes the whole ensemble of firing rates in the corresponding control ensemble. Okay, so this part was done with several collaborators, mostly at Carnegie Mellon, pictured here, and then Cati Vich from Mallorca. So this is part of a CRCNS award that Cati, Tim, and I have along with an experimentalist, and we're still doing lots of follow-ups. This is kind of the polished part that we have. Okay, and so now, what do I have, like a few more minutes or 10 more minutes or?
Few more minutes.
More minutes. Okay, so let me just mention a little bit that we've done on plasticity. Okay, so there is, as I mentioned early on, the dopamine here in the basal ganglia, which is has a huge impact on the corticostriatal synaptic weights. Okay, so now that we've got all of this baseline in place, what we want to do is, instead of having ourselves, tune these basal ganglia weights, we want to actually let the plasticity play, and we want to have conditions where there are actual rewards, right, or simulated rewards, and like better choices and less optimal choices, and see how different reward contingencies actually push the weights, and see how that relates to the control ensembles that we predicted should be affected. So this is in progress. I put this up here, and I will talk about this with, I guess, the way we set up this corticostriatal plasticity with dopamine builds on a variety of past works. A couple are listed here, and then there are earlier ones that those build on, but the idea is with each of the choices that you can make, you have a reward. Okay, you have a representation of the value of each action, and this is a standard way to update that. It's called Q-learning, right? So you basically take the difference between the reward and the expected value and use that to update your representation of the value. Okay, then what we're going to have is a dopamine signal that corresponds to how the reward compares to what we expected based on the available values in the task. Okay, and then based on the dopamine released, we are going to have, we're going to have a weight update. Okay, but which weights get updated can't just depend on dopamine, because this dopamine signal is common to all the neurons. Okay, there's also an eligibility component, and there's a lot going on in this eligibility component that I'm not going into here. Okay, the eligibility component is based on the timing of the presynaptic, so the cortical spike and the striatal spike. It's an SDDP component that sort of determines at any moment who's eligible to have their weight updated. And then the weights go into updating the synaptic conductances. Okay, so when we implement this sort of three-component, three-factor, cortical, striatal, and dopamine SDDP, we can do a couple of different things. One thing is we can look trial by trial at how the reward, how the outcome of a trial, whether we get a reward or not, moves us in the DDM parameter space by doing this fitting process. And so these are some very early results suggesting, the initial rewards tend to increase the drift rate, negative outcomes especially increase the boundary height. And so we'll be continuing to look into that more deeply. And then over the long term, once we sort of saturate at a certain performance level, right, you can see these are the changes in accuracy, reaction time, and then these two corresponding DDM parameters that result from this plasticity. And what we see is our accuracy does go up as we would want, Interestingly, beginning to end, the reaction times don't really change much. And correspondingly, we really see a lot of change in the drift rate without change in the boundary height over the long term. So the plasticity is effectively tuning the drift rate. And if we can do a CCA analysis here, and sure enough, we see that the components, I'm just skimming this bit, the components that kind of show up as being different due to this plasticity are exactly those difference components, and that showed up in the choice ensemble from our earlier analysis. So the upshot here is that this synaptic plasticity at this corticostriatal level seems really well tuned to adjust the choice ensemble. But then that leaves open a question of, well, it looks like we should have at least 3 ensembles to effectively tune our decision policy. So what other tuning mechanisms could be present in the basal ganglia besides this plasticity? Okay, and so what I won't tell you about is one example of something we think could be doing this. And so because I'm out of time, I'll just point you to this paper. Okay, so if you're interested in another possible tuning mechanism that's different from the traditional dopaminergic plasticity that could be present in the basal ganglia, we did this paper which came out in 2020 already, and it's taking us a while to complete ongoing follow-up, but it's in progress, where we modeled the output neurons of the basal ganglia. So here I've switched from GPI to SNR. But importantly, we included the chloride concentration in these neurons. Okay, and so one of the things that could modulate activity in these pathways is remember, there is so much firing happening, right, in these outputs. Okay, when there's firing at high rates, you can actually start to change the ion concentrations. When you change ion concentrations, you can change reversal potentials. Okay, and so we have a theory about how that could actually, by modeling the chloride concentration, we can look into this idea that could actually help tune some of these decision thresholds in the basal ganglia. All right, so with that, I will stop. And thank you for your attention, and I'm glad to take questions.
Thank you, Jonathan. Do you have any questions?
Paul,
nice talk. I like the mapping between the biology and the math equations. So I have a couple of questions. So one is, it's been quite a while since I've been working on this, and you basically having the basal ganglia essentially do everything. The symmetry breaking and so on occurs there, not in the cortex. Yeah. Whereas I kind of recall like LIP data was already showing symmetry breaking at a fairly early stage in cortex. So how do those two square up?
Okay, so there's kind of 2 approaches to answering that. Okay, so one would be to try to be like the really strongly, like it's all basal ganglia approach, which is that there are experiments where people, manipulate cortex in such a way that it's not doing that sort of symmetry breaking, and you can still get, this action selection effect, apparently just through the basal ganglia. You also do see the ramping in striatum. Okay, so that would be one answer that some people could give. I don't fully believe that. I believe that it's like a teamwork situation, right? So the cortex will be experiencing some symmetry breaking based on the evidence for different options, and that gets projected down to the basal ganglia, and then the basal ganglia plays a role in sort of allowing this to go ahead and modulating decision kinematics, right, or action kinematics if it's a movement based decision, and also sort of drawing in experience, right? So the cortical symmetry breaking may be driven by perception, but if you already know that there's been bad outcomes associated with one decision, or good outcomes associated with one decision, that'll be represented in those corticostriatal weights. And so despite the presence of a symmetry breaking in the inputs, the basal ganglia weights could sort of override that and end up at the very least shifting your reaction time, but probably also changing your choice distribution. So I believe in that sort of synergistic view.
Okay. And I guess my second quick question. So I recall a paper, I think Eric Shea Brown was first author, where they suggested norepinephrine, a neuromodulator, would control the speed accuracy trade-off by essentially boosting excitatory synaptic strengths. Have you looked at that as another way of like tuning your parameters?
Yeah, we have not. I mean, that's exactly the type of thing we're after, in that idea that there should be some other tuning mechanisms besides, the dopaminergic plasticity. So that would be a great candidate to look at. I agree. Thank you. Yeah.
I have a brief question, I guess. So I was wondering, I mean, I found it very interesting that you find this mapping between function and the parameters of the network. I was wondering how important was that to use spiking neurons, and if you could have similar effect with, I don't know, with a network of Wilson-Cowan equations, and in that case, would you be able to say more analytically about the?
So the reason we went with spiking is because we want to be able to have the STDP, right?
Yeah.
So spike timing dependent plasticity. So we need the exact timing of spikes in individual neurons. And so now, the way we have things set up, only those, say, ISPN neurons that were active, within a reasonable time window of a decision, are going to get their weights changed on that particular trial, and likewise for the DSPNs. So, excuse me. So I think, even though it wasn't a real part of most of what I presented, having the plasticity is completely fundamental to this project. So that's why the spiking. Yeah. So in some ways, this is the result. This is a project where we just said, look, doing actual, like, proving anything about this, or more under the hood mathematical analysis is completely on the back burner. All about getting the answers that we want and getting the biological insights that we want. And I think that should be okay, right? I mean, there's some situations where you really want to do analysis, and others where it's just not the priority.
No, for sure. Yeah. And then another question I had is, what happens if you had overlap on the networks then, with cells shared between channel A and channel B, or if you had more choices in the sense?
All right. So, oh, by the way, if you had more choices, that's usually the big first question. Right? Like, when do you actually have two choices, right? Like, no menu has two, well, very few menus have two foods on them. Okay, so interestingly, there are some results that suggest that even when we're making decisions among many, many options, the way it works is actually a large sequence of two choice like mini decisions. So that says, two choice is a good thing to study. However, yes, there are frameworks that allow, like, generalization of DDM or other, other representations of more than two choices, right? So yes, that should be, a natural thing to try, but it just makes things more complicated, and we don't feel ready. We don't feel there's a justification for doing that quite yet. The first part, say again, I got, what was the other part of your question?
It's related, but it's, what if you had overlap in the neurons you use for channel A and channel B?
Yeah, that would be more realistic. No one knows, I don't know, the details of the level of overlap or whatever, but surely they aren't perfect pure action channels. But No, it should be fine. You know, that's going to add a sort of noise. A really interesting problem too would be like, can you form these action channels in the first place through like reward-driven plasticity?
Right. Do you create habits also? I mean, by providing repeated.
Right, and what happens when you face a new stimulus that's sort of in between two that you've encountered before? You know, yeah, there's lots of interesting questions there, I think.
No, it's a great model. I like it a lot.
Thank you. Yeah. Our collaborator, Aryn Gittis, and his student, Julia Bedina, are doing some really interesting experiments where they can stimulate, you know, these two different striatal populations during decision-making, these mice and these Y maze tasks. And yeah, it's going to be fun. We were just talking about that earlier this morning. There's a lot of interesting results there.
And so another question that I had, like it, which is theoretical in a sense, but I mean, we know exactly what the first passage time of a, you know, of a Brownian motion looks like. But do you find, and I agree that you can feed some parameters of it, but does the distribution you get from the spiking network look like, and how much does it look like the first passage time? I mean, if it makes sense.
Yeah. Yeah, so the reaction time distributions that we get can be fit really, really well by the DDM if we, you know, give ourselves freedom to choose those 4 parameters. So, you know, coming up with a mathematical argument that this model sort of implements something DDM-like in a more literal way seems like it would be a really cool result that I have no idea how to do. And yeah, if you had other thoughts on interesting mathematical directions related to this, even though I said it's on the back burner, I'm certainly interested in those things.
But the DDM itself is maybe not the gold standard. I mean, it's a very simplified model. It's a very linear model in a sense.
Super convenient for this purpose.
Right.
And because our decision threshold, you know, our sort of decision, the way we have decisions implemented in our network is pretty simple. At the end of the day, even though all this action happens, you know, it is a hitting of a threshold by a firing rate. So in that sense, the DDM is kind of a good match. You could say by design.
Maybe just one comment, if you do want to go in the more mathematical direction and keep the STDP, there is a way of incorporating it in firing rate models. If you assume Poisson output, then you can use the statistics to say how much, you know, how likely you are to get a spike pre, before, post, or vice versa. I think Pfister has a paper doing that. So there is a way of incorporating it, but it might be more work than it's worth.
Yeah, we are looking at some of the properties, mathematical properties of that STDP rule or the dopaminergic STDP rule that I put up on the screen, but that's at a really early stage.
I don't know if there is just a enthusiastic comment from the participants, but I don't know if there is any more question.
Do you have more questions? If not, let's thank Jonathan again.
Thank you.
Thank you.
Yeah.
Nice seeing everyone.
Good to
Hope
see you.
to see you in person in the near future.
That would be great. And yeah, John, I'll be in touch at some point, or you can contact me if you think the time is right.
Sure. Yeah.
Yeah. All right.